Today, I will present the discovery of a new aging mechanism and medical implication using this discovery to rejuvenate DNA. This cartoon shows the new aging mechanism that in our DNA are composed of DNA gap. The function of DNA gap is to release the torsion force when DNA denature. The denature of DNA occurs when DNA work, just like transcription or DNA replication, and then the double helix structure of DNA will spin on DNA or the length of DNA. However, without DNA gap, the denaturation will cause the torsion force or the length of DNA, and DNA can be damaged easily. In young DNA composes a lot of DNA gap. That's why we call it youth DNA gap. In old DNA, the number of DNA gap reduced. That's why DNA of the old can be broken easily. We also discovered which protein produce DNA gap, and we call the protein molecular systems. The discovery of DNA gap was reported in 2008. The discovery of molecular system was reported in 2013. The role of DNA gap in stabilizing genome and reduced when DNA age. It was reported in 2018. Today, I will present the current work that if we introduce DNA gap into cells, we can increase their durability and also rejuvenation. In 2008, we tried to measure the methylation status of endogenous DNA double-strand break, which is we changed the name of DNA break into the DNA gap correct in the current paper. In the past, we measured the methylation of the DNA gap, and we hypothesized that the methylation of DNA gap is supposed to be hypomethylated. But the results show that every sample of every cell, DNA methylation of DNA gap were hypermethylated. The x-axis represents the methylation of the genome. The y-axis represents methylation of DNA gap. If DNA gap and the genome DNA methylation are equal, or the DNA methylation data is supposed to be here. If the DNA gap methylation status are hypomethylated, then the methylation status is supposed to be here. So what we have is opposite to our hypothesis. Our hypothesis thought that DNA methylation of DNA gap is supposed to be hypomethylated because in hypomethylated genome, there are phenomena we call genomic instability, and mutations such as DNA deletion happen easily. So what we detected is opposite to hypothesis. This is a serendipitous discovery, and usually when you have serendipitous discovery, this is important information. We also detect DNA gap in every phase of cell cycle without and not relying on DNA replication. So the modification of DNA gap are not the same as what we thought, that they are DNA damage. So if they are not DNA damage, they are supposed to be epigenetic marks. If they are epigenetic marks, they're supposed to be produced by cellular enzyme and also has physiological functions. So in 2013, we tried to search for molecular systems, and by measuring DNA gap in yeast that has deletion of one gene, and if one of those genes has reduced DNA gap, then the function of the gene has a role in producing DNA gap. And we find two groups of genes. One gene is SIR2, the other gene is HMGB group. SIR2 is quite interesting at the time because SIR2 is SIR2 in human, and both SIR2 and SIR2 has been studied that the role of aging process. So we can make a hypothesis at that time that DNA gap has a physiological role in preventing DNA aging process. For HMGB group, we think that this may be the molecular sister, particularly in NHP6A, because NHP6A in human is HMGB1, and there are studies showing the association or the function of HMGB1 with physiological DNA break. Also, in conclusion, in 2018, we studied the role of DNA gap. And the role of DNA gap is to increase DNA durability, as I explained earlier. In our daily life, we can compare the role of DNA gap with the gap of railway train, that in high temperature condition, the train track will be bent without the gap, this small gap. This small gap will allow DNA will allow the track of the railway train not to be bent. So in 2018, we proved that DNA gap reduced in aging cells, and the reduction of DNA gap caused the DNA tension and caused DNA to be broken. In conclusion, this is a DNA aging theory that people know that if you have DNA damage will send a signal transduction called DNA damage response to stop cell division and cause growth arrest. A persistent DNA damage response will change anatomy and physiology of the cell to senescent process. But people do not know how aging DNA accumulated DNA damage. In this paper, we proved that reduction of DNA gap will promote DNA tension and cause DNA damage. So we come to this paper that we try to introduce HMGB1 to produce DNA gap and see what happens. Before we study, after we study this, we study human, rat, and senescent cells. We study naturally aging human, and we see that the more human age, the lower DNA gap. We also follow up the same individuals for 4 years, and we can see the reduction of DNA gap after they increase age. We also observe the reduction of aging rat, naturally aging rat, and the rat that we introduced aging by the galactose. We also observe the reduction of DNA gap after we induce cells to be senesced by several chemicals. In conclusion, DNA gap are reduced in all kinds of aging cells, both naturally occurred or chemically induced. So next we study the composition of DNA gap, and as our hypothesis, we prove that the DNA gap are produced by HMGB1 proteins in the box A part. HMGB1 protein composed of box A, box B, and N terminal. We prove that box A is the molecular sisters, and histone acetylation prevent histone deacetylation prevent the DNA gap to be recognized by PDR, and histone deacetylation occurs by 30 months. We perform 1, 2, 3, 4, 5, 6 experiments to prove this mechanism. This slide shows that if we inhibit histone deacetylation by chemical method and genetic method, we can the DNA gap that produced by box A will be recognized by H2AX, which is the primary DDR. So we study the function of DNA gap. You can see that first we measure endogenous DNA damage and endogenous DNA damage response. After we introduce box A into cell, the endogenous DNA damage and endogenous DNA damage response were reduced. And then we introduce the box A into cell to produce DNA gap. And then we brought these cells to be irradiated, and the radiation, if the radiation damage cell, we can detect the DNA damage by measuring H2AX size, or measure the degraded DNA by comet assays. And you can see that after we introduce box A into cell, the cell cannot be damaged by radiation. So this is quite important because this is the only technology in the world that can increase DNA durability, and maybe the only technique in the world that can prevent radiation toxicity. And under current situation, we really want this medicine. Okay, then I prove that the increasing the durability by box A is because of DNA gap. We perform 2 experiments to measure that around DNA gap there are no DNA damage. In the previous slide, we showed that if we introduce DNA gap by box A, the number of DNA damage in the whole genome are reduced, and also increase DNA durability. In these 2 experiments, we proved that around DNA gap, the DNA damage that reduced is actually most frequently occur around DNA gap. So in these 2 experiments, we proved that the function of DNA durability increased by box A is because of the produced DNA gap. Then, because if you remember, we know that DNA damage can induce senescence. DNA gap can reduce DNA damage. So if we increase DNA gap in senescent cell, we should be able to rejuvenate cells. So in this experiment, we introduce cell to be senescent by etoposide, and then we transfect cell with box A using other plasmid as a control. This 3 plasmid is also box A, but has mutation at the key amino acid or molecular systems. You can see that only box A can rejuvenate cells, but other plasmid cannot rejuvenate. And we also show the cellular structure of cell, show the number of SA beta, which is senescence marker. We also show increasing DNA gap, and we also show the increasing cell viability by box A. So we should be able to, after we be able to rejuvenate cells, we want to see whether we can rejuvenate rat or not. We studied 2 models of aging rat. The first one is the galactose-induced aging model. This one is similar to aging by sugar, because this is a toxic sugar, just like diabetes. The other group is we study natural aging rat. and we measure the liver function. You can see that liver function improved back equal to young rat. This also show liver function back equal to young rat. We study the number of senescent cells, and you can see that the box A can reduce the number of senescent cells and also reduce visceral fat size. We study learning and memory. Memory go back to equal to young rat, but learning is quite strange that it's not only better than control group, it's also better than young rat as well. I have a hypothesis, but I don't have time to explain. Okay. The other group, we measure other proteins in liver and in brain. And you can see the reduction of DNA damage associated or senescent associated protein in rejuvenated rat. This shows the confirmation of reduction of senescent cells, that these are livers of young rat, aging rat with box A transfection, aging rat with fake box A transfection. You can see the reduction of SA beta positive cells in aging rat liver after box A transfection. This one is quite important because not only we can rejuvenate cells, we also be able to rejuvenate extracellular component as well. That this slide shows reduction of fibrosis in both models after box A transfection. This is quite important because we thought that fibrosis causes stasis, but actually after we can rejuvenate cells, we also be able to rejuvenate extracellular component. This has an immense clinical implication that, for example, we should be able to reduce amyloid in Alzheimer's brains. This also shows the change in histology structure of rat after rejuvenation. In conclusion, we think that the new aging mechanism is beginning with HMGB1 release, because if you the literature show that when cell begin to age, HMGB1 will release out of cell, and the reduction of HMGB1 intranucleus HMGB1 will cause the reduction of DNA gap. The reduction of DNA gap will increase DNA damage, and the increased DNA damage will increase DNA damage response. Because it lacks DNA gap, the DNA damage response persists. Then cell can enter cellular senescence. So using box A as a molecular sister to produce DNA gap, we can stop this process, maybe permanently. I call this technique as an upstream senolytic activity of box A produced DNA gap, because you can see that DNA damage alone can induce variety of molecular phenotype and physical phenotype of senescence, so that when we can inhibit DNA damage, then this is the upstream activity of senolytic. In conclusion, we be able to rejuvenate rat and also reduce liver fibrosis. In addition to the rejuvenation work, we also test the box A component in treating burn and also DM wound, and we also test to safety using pig. And in addition to safety test, we can see that the pork of those pigs meat quality of pork much better, both soft and also firm. So that we can conclude that by producing DNA gap by box A, we can protect DNA. This also a new term because in the past people do not know DNA protection. The method that people know that when cell can reduce DNA damage is DNA repair. This is additional mechanism that we can prevent DNA damage by DNA protection. And the DNA protection function can lead to medical application by prevent DNA damage, such as radiation toxicity. I hopefully that this is not necessary, but under current situation, we are at risk of nuclear war. And if it occurs, bauxite or REDGEM is the only hope. It is only medicine that have potential in reducing radiation toxicity. Because of DNA protection effect, we can inhibit the senescent process at the beginning of the senescent process, so that this DNA protection effect is upstream senolytic that can rejuvenate cell completely, rejuvenate rat completely in the whole body. Also rejuvenate, in addition to rejuvenate cells, we also rejuvenate extracellular components. So this property of REDGEM can be used, has a potential to use to treat age-associated diseases and conditions. In short, REDGEM represents the rejuvenating DNA by genomic stability molecules, i.e. D equal to red, GEM equal to gems. That's why in Thai we call box A produced DNA gap as a money bank. ขอบคุณครับ